properties of the drug is very much important for the better dosage form preparation or formulation, right? Because we know that maximum, at least 70% of the drug molecules are practically very much low solubility in aqueous environment, or you can say insoluble in aqueous environment. But as our body, human body is full of aqueous medium, so the solubility issues is very, very crucial for to get its proper bioavailability. So from this side, solubility, as we know, that it is a summation of two properties. One is called lattice energy and one is called hydration energy. That means, as we know, that heat of solution is equals to heat of, that means lattice energy plus heat of hydration. So that means first, the drug molecules, for its stability, crystalline nature is preferential as compared to amorphous, because generally amorphous form is not stable. But although in today's era, maximum work is going on to stabilize the amorphous drug delivery system. That means if you can prepare the amorphous form and if you can stabilize it, so the maximum benefit we can get it from this thing. In other way, that co-crystal is very crystal, its definition is very beautiful definition. This is the most non-living ordered substances. That means living body is the most ordered, right? So if we just exclude living body, then the non-living within the circle of the non-living system, crystals are most ordered substances, right? So just okay. So if we say that is the reason behind the, if we find that if we would like to get a stable drug molecule, so always we try to get the crystalline property. So crystalline property means what? That means the molecules are highly ordered, right? In amorphous, they are highly disordered, and due to these disorderness, their solubility will be high, and due to the orderedness of the, or high degree of orderedness of the crystal, the solubility of the drug will be less, because the crystal first, crystal has to break down to its molecules, then that molecule will interact with the solvent, and that solvent will form a solvent seed, or you can say that just like a seed, so that they will be in a solution, right? So this is the basic principles of the how a crystal material will go into the solution. So co-crystal, it is basically, it is nothing but the one single crystal lattice containing two or more components. If you say that what is molecule? Molecule is all about atoms, ions, and covalent interactions. And what is supermolecule? Supermolecule means molecules and non-covalent interactions. Non-covalent interaction means mostly hydrogen bond, van der Waals forces, pi-pi interactions, steric repulsive forces, all is under the bracket of non-covalent interaction. Definitely they are weak forces. So if we have a supermolecule, so crystal is nothing but a supermolecule, because crystal is being formed by the intramolecular interactions of the more than one molecule. So then only crystal will form, and if the molecules are arranged in an ordered way, they will form a specific geometry, and that will be actually the crystal. So now co-crystal is what? Co-crystal is basically nothing but it is a single crystal lattice. That means it's a single crystal containing more than one component, right? Just like if we say that hydrate is also like that. Hydrates is because when water molecule participated in the crystal formation, then we know that is called the water for crystallization, right? Because that water is required for the crystal structure or crystal lattice. So if we have the option to prepare a single lattice containing more than one component so that we can manipulate or we can fabricate its physicochemical properties. Mostly, what are the different physicochemical properties of the drugs we can change by tailoring the co-crystal or by synthesizing the co-crystal? We can, most importantly, we can change the solubility pattern, right? Solubility. Hello?
Yes, sir. Are you changing the slide? Because
No, I'm not changing. I'm giving the introduction. Okay.
Sorry, sir.
Okay. So, what are the different properties actually we can change or we could modulate depending upon our need or intention? So that is called, it is called tailoring method. Tailoring means we can make as per our wish. So here, if we say that a drug, it is having an ordered crystalline structure, due to its crystalline structure, it will show a very poor bioavailability or due to its poor solubility. So in that case, if we can incorporate a second material to its crystal lattice, so that crystal lattice will be definitely a new crystal. It will be neither drug crystal or it will be neither the second component. Second component here it is called the coformer. So it will be a different crystal lattice, and that crystal lattice will show improved solubility, improved dissolution rate, basically intrinsic dissolution rate. It can also modulate the permeability. It can also modulate the photosensitivity. It can also modulate the moisture absorption capacity, that means hygroscopicity. It can very much, it can modulate basically the tabletability property, that means compression and compression, that means tabletability means compact compression, tabletability and compactability, that is called CTC property. That means mechanical property also we can tailor by incorporating the coformer into a new crystal lattice with the drug. So because why I am giving saying all those things, because what we will discuss, that will be a particular case. So before that, we must have some introduction. That's why I am discussing this thing. That mostly there are different hypotheses by means of which we can say that how a co-crystal can change the solubility. Either it will increase or it will decrease, right? So it absolutely depends on the second component, that coformer what we are using. So most of the work till now, it is basically, it's the work started in the late 90s and in the early this century. So it actually, Professor Desi Raju coined the first terminology, what is crystal engineering. Because engineering means there must be some design, right? There must be some prediction. So similarly, if we can make a crystal according to our intention, according to some design, according to some prediction of their property, so that's why its terminology is basically known as crystal engineering. The process is known as crystal engineering. So after that, huge amount of work on pharmaceutical, mostly pharmaceutical field, it has extensive work. But one confession, till now very few co-crystal products are in market. One component it is in market by Novartis, that is for valsartan sacubitril for better heart disease control. Recently in 2020, JISU-PHARMA of Spain got the US FDA approval for one co-crystal. That is, you can say that is a great thing. That is basically your tramadol and celecoxib. Tramadol and celecoxib. This is drug co-crystals. They got the US FDA approval for marketing. Apart from these things, there are multiple antidiabetic groups, that is ipragliflozin, canagliflozin, all these things have already in market as a co-crystals with coformer. So if most of the work, if you see in literature, then all the
Hello. ওনার Hello, sir. I think sir, network connection is poor. Participants are requested to stay online. There was a technical glitch, which is why our speaker has left the meeting. He is joining soon. Just be online, please.
Sorry for the disturbance. Actually, I was disconnected. Okay.
Yes, sir. You are now audible and visible.
Yeah. Okay. So, if we say that most of the work on pharmaceutical co-crystals, which are in literature, they basically tried to report the solubility aspect. That means a drug is a low solubility, but they would try to improve their solubility by crystal engineering. So our work is basically just opposite. Though very few works were reported, that if we have a very highly soluble drug molecule, so what we can do, because if we know that if we have a very high soluble drug molecule and if we have a very short half-life, so in those cases it will be better to prepare a sustained release dosage form. So here if we have used a drug molecule which is known as pirfenidone. Pirfenidone, it was approved in 2014 by US FDA for the treatment of COPD, right? All of you know what is COPD. But the dose is very peculiar and it is highly soluble. Its solubility is approximately 20 mg per ml. And its dose is like that 2 gram 400 milligram, or better to say 2403 milligram per day, its dose. That means very high dose, right? And its dosing is also peculiar. Dosing is for first 1 week. It's actually this drug, as COPD patient will be taking this drug for the rest of his life or her life. So body has to acclimatize with the drug. So what is the procedure for this dosing? First, 200 mg as per Indian concept. 200 mg, the product is known, it is by Cipla. 200 mg thrice daily, it will be continuing for 1 week. And then 200 mg thrice daily, then 400 mg thrice daily for another week. So in that way, up to maximum 2403 mg per day. So our aim was that if we can prepare a sustained release product by crystal engineering technique, as we know that if a drug is poorly soluble, there is no need to develop extra efforts to develop the sustained release formulation, because sustained release means we are basically controlling the release by incorporating some polymer which will control the release and which will basically release the drug in a sustained fashion. So similarly, if a drug is insoluble, if you see that most of the drugs which are insoluble, we do not find their sustained release dosage form because their solubility itself acting to retard the release or to control the release rate. So our hypothesis was like that if we see here that pirfenidone, it is a highly soluble drug, and we would like to develop a sustained release formulation by using the crystal engineering technique, and there we have studied the viability study, all these things. So first of all, co-crystal, you need to think that is basically new things in terms of what if you have a new drug, you need to do all the steps to develop the formulation. Similarly, a co-crystal also you need to consider that is that like that you have a co-crystal, as co-crystal is basically the play of non-covalent interactions. So there might be a different scopes where that non-covalent interactions will break down. So if it is break down, then what will happen? Then it will be converted into drug. So then we won't get the benefit out of the co-crystal. So there are 8 stages majorly, if we say. First thing is the coformer selection. That is most crucial because this is the only thing by which we can tailor any physicochemical properties. Say if a drug is A, if we use B is coformer and C is another coformer, the properties of the co-crystal will change because B and C, that will basically inculcate the properties to the drug molecule so that we can get the benefit out of it. So that's a crucial step, that what would be our coformer. Generally, the coformer in case of pharmaceuticals, it has to be from the GRAS list. GRAS list means generally regarded as safe list. So, and there are some rules. One is called del pK rule. That means the difference of pK of the drug and coformer. Another one is called crystal structure database screening rule. So what are the different co-crystal has been formed by which this thing coformer, that screening we can do. And most importantly, that is from the structural analysis, that is supramolecular synthesis approach. So by using all these things, nowadays there are many calculation based also is coming. CosmoRS, there is one software where we can calculate some thermodynamic parameter by means of which we can say, okay, there might be a probability to form a co-crystal with this particular drug. Second one is co-crystal formation. Yes. Hello, can you hear me? Hello. So second step is after screening the coformer, we will prepare the co-crystal.
Yes, sir, you are audible.
Yeah, just I am hearing some noise, that's why I am stopping. Okay, so co-crystal preparation, the most simplest is the solution crystallization method. But solution crystallization method cannot produce. Hello? Can you hear me? Someone is talking, please.
Yes, sir, please go ahead, sir.
May I continue? Hello.
Sir, please continue, sir.
Okay. So co-crystal preparation, solution crystallization technique, slurry method, then milling method. There are different methods. Stage 3, we need to characterize co-crystal. That's a very tedious job. If you get the single crystal, That is the best way to characterize the crystal. That means if you can have a single crystal, you can go for single crystal XRD, and then you can get the powder XRD, DSC, TGA, FTI, Raman, all these things are supporting if you have a single crystal data. After getting the single crystal characterization, we will go for the property evaluation. That means what is its solubility, intrinsic dissolution rate, permeability study, mechanical properties, stability issues. Then we go for the pharmacokinetic study. If say our aim is to increase the solubility and we got the enhanced solubility, then we should expect that its bioavailability will be increased. So we will go for the bioavailability studies. And after this is basically all our pre-formulation steps. After that, we need to do the, this is the, up to this thing, it is basically the development stage of the co-crystal. And then we will start for the formulation stage. Before going to formulation, we need to develop the pre-formulation studies. So pre-formulation studies is very, very important because first and foremost thing is its drug excipient. Because if you prepare some solid dosage form, say for example tablet, we need to have some excipient. So our aim is to use some sort of excipient. Those excipient will not hamper the non-covalent interactions of the co-crystal. If you say many polymers, because most of the polymers, all of the polymers have a lot of OH group, so they may interact with the drug molecule or coformer. So if they will interact, and then entire thermodynamics will change, and there might be an option that where the non-covalent interaction will break down. During mixing, during milling, during compression also, because compression one step is rearrangement step, right? So in there also there might be a chance. So we need to prove in every stage from where we can, there is a source of breakdown of the crystal lattice. So that we need to very much important, right? Say for example, if which type of granulation technique we will use. Is it wet granulation? If it is wet granulation, then we need to do the stability studies. Because in stability studies at higher humidity, whether that co-crystal, that non-covalent interaction is breaking or not, that we need to confirm. After that, we need to do the scale-up process, then regulatory applications. So these are the pharmaceutical co-crystal preparation and characterization. There are three methods mostly. One is called solution-based methods. So most important thing is that evaporative crystallization, that is also known as solution crystallization. Next one is reaction crystallization, cooling crystallization. These are all solution-based. Solid state method, hot melt extrusion of co-crystal, it might be, because in hot melt extrusion, if we can melt both drug and coformer and allow them to crystallize, then there might be an option to get the co-crystal. And mechanochemical grinding, that means if we grind it in a ball mill or in a high-speed mill, in that case we can get this thing. There are several other methods also, that is called spray drying method, anti-solvent crystallization, freeze drying, ultrasound co-crystallization, supercritical carbon dioxide processing, resonance acoustic mixing, and ionic liquid crystallization. These are the methods, but generally in our lab, we used to follow two things. One is mechanochemical grinding, and another one is evaporative crystallization. And more in sometimes we can also use the reaction crystallization. So what are the different characterization technique? Most important characterization technique is X-ray diffraction. In X-ray diffraction, there are two things. One is single crystal X-ray diffraction, one is powder X-ray diffraction. So most importantly, if you like to characterize co-crystal, so the best way to get the single crystal and characterize by using the single crystal X-ray crystallography. DSC, spectroscopic studies, that is FTI, Raman, all these things, hot stage microscopy, and efficient field emission scanning electron microscopy. So co-crystal preparation is very, it's now its theory, its procedure, we can, it is established. But formulation is very tedious, because in each and every step, there might be a scope to break the lattice energy. So if the lattice energy of the co-crystal breaks, then although the co-crystal having a good solubility property, good mechanical property, but we could not prepare the formulation. The most important thing is its solution phase stability. That is called solution mediated phase transformation. Say for example, if we put co-crystal in any solvent, mostly aqueous solvent, if after giving within a minute, if that co-crystal breaks and it has been converted into drug and co-former, then that type of co-crystal, they're very much challenging for the formulation development. It's basically very much challenging. But if it retains its crystal lattice in the solution at least for 1 hour, that is the best ideal situation for the formulation development, right? So if we say this is the molecular things, that is crystallization, that is co-crystal, this will be the structure because two molecules will interact through hydrogen bonding. And this is the formulation. We need to do the blending, then we need to do the compression, all these things. So what are the formulation challenges if we say? Formulation challenges, if you say co-crystal, then if we add excipient, if we just process it, then we will get the tablets. But the problem is the hydrogen bond propensity, because excipient, they may form, because we know that binder, basically the binding activity of excipient or any binder, in that particular case also hydrogen bond plays an important role, right, with the drug or with the other excipients. So that means there is a possibility of hydrogen bond formation during the granulation process. So that means whenever the hydrogen bond already present in the co-crystal, and again during granulation, if there is a possibility of forming the hydrogen bond, so there might be a competition with which actually the hydrogen bond will form. So that is why that co-crystal excipient compatibility study is very much important for the formulation. Then another thing is surface activity, because say excipient is basic in nature or acidic in nature. So in that case also, that surface pH or microenvironmental pH, all these things will play during the granulation technique, and ultimately if we won't get the after tablet, if we won't get the co-crystal its intact form, there will not be any profit out of this thing. So during milling also there might be a scope or there might be a chance to break the hydrogen bonds, right? So if we just go to the why we have taken this approach, that if we can reduce the solubility of a drug molecule which is highly soluble, so we can get the sustained effect. That is our hypothesis. If we just back to our theoretical consideration of the Noyes-Whitney equation, we could probably understand that if we see this is the solid and this thing is basically the boundary layer, right? And here there is basically two-step process in diffusion mechanism. This is basically the mass transfer phenomenon, and here are two steps. One step is from the drug or core of the tablets or core of the matrix, drug will come at the surface, and at the surface they will form a saturated layer that is called boundary layer, and from that layer the drug will diffuse to the bulk of the solution. So the concentration of the drug at the saturation layer or boundary layer, that is basically constant, because when that is basically the reservoir, it acts as reservoir like that if the sum of the drug is going to the bulk, so there will be a vacuum. So from the matrix, drug will come to this thing. So that is nothing. It is one type of apparent zero order rate kinetics. If you see that always the concentration of the drug at the saturation layer or stagnant layer, if you rather to say, so that is constant. So that is what? That is nothing but the saturated solubility of the drug. So from this equation, we all know this equation, dC by dt is equals to dS, dH, Cs minus C, all these things, surface area, diffusivity, thickness, stagnant layer thickness, and the concentration. In case of polymeric matrix, what we generally do, we will increase the H value. right? Because we have given some polymer that will soil. Due to soiling, the path length, that means the length or thickness of the boundary layer will increase, and so drug will travel that path. That will take time, and accordingly the drug will be slowly released. But if we say that if we can manipulate the CS minus C, that means CS, because C is the bulk concentration, but that means if we can change the CS, then also we can control the dc by dt, or you can slow down the by dc by dt. dc by dt is the rate at which drug is going to the in the bulk from the core or matrix. So that is basically the fixed law, nothing else. That if we see that fixed law is, if we can control the CS, that concentration gradient, if we can reduce it, so dc/dt will be reduced, and if dc/dt will reduce, that will act as a sustaining action. That will basically the sustaining action. So our aim was, if we can reduce the CS, and CS, how can we reduce? Because saturated solubility generally it depends upon the temperature, it depends upon all the solvent, all these things. But in a particular solvent at a particular temperature, we can change the saturated solubility by changing its crystal lattice, because it is highly dependent upon the crystal lattice energy of the particular molecule. So if we can change it, so then we can get this thing, it will be reduced, and if it is reduced, then dc/dt will be reduced. That is basically the theoretical hypothesis of our work. So we have a pirfenidone which is highly soluble. If we reduce its solubility, that means we are trying to reduce its CS, saturated solubility. So therefore we can reduce the concentration gradient, and followed by, so it will definitely lead to get the reduced diffusion rate. So that is our theoretical consideration. All these things background we have already discussed here. So now if we just trying to discuss the hello, how much time may I get? Hello, can you hear me? Hello, hello, can you hear me? Hello. Hello, can you hear me? Okay. So now if we have, if we try to discuss the preparation, that means how we have proceed. So the basic fundamental is that if we would like to reduce the saturated solubility of a drug which is highly soluble, so we must be using a coformer which are less soluble coformer in water. So we have used many carboxylic acid. We have tried turmeric acid, pimelic acid, trimesic acid, oxalic acid, glutaric acid, malonic acid. This is a thumb rule, but that won't work always, right? So fortunately, we got two co-crystal with fumaric acid and trimesic acid, right? If we see, this is the DSC thermogram. Obviously, we know that DSC thermogram, what will give? So if genuinely co-crystal is formed, then what will happen? Then the peak melting temperature will be different. It will not be similar to the either drug or coformer. It will be a different one. So if we see here, in case of, this is the peak melting temperature of the pirfenidone, this is the peak melting temperature of the fumaric acid. But when we have got the co-crystal, that is PFDFA, is mixed F. So PFDFA, we are getting this thing. If you see, these are not same. It have a minimum of 7 to 8 or 9 degree difference, right? And in case of trimesic acid, if we say this is the pirfenidone and this is trimesic acid, so we are getting the sharp endotherm at in between. So this is the new, that means it proves that there must be some new crystal, right? So, and that is the reason that why its peak melting or endotherm or peak melting temperature has changed. So this is nothing but its corresponding thermogravimetric analysis, that it gives the information about at which point, at which temperature range it starts to degrade and its degradation completes. So one thing from the thermographic analysis, we can confirm that melting is not accompanied by degradation, because degradation starts after melting. If we see that approximately after 150 or 200 degrees Celsius, it starts to degrade. That means this is the phase transition. This is not the decomposition. In many cases, the decomposition might be giving the endotherm peak, although general decomposition will give you the exothermic peak, but in many cases it might be giving you. But to confirm this thing that this peak is exclusively for the phase transition, that means solid to liquid, so we need to do the thermographic analysis, so from which we can say that yes, it is your this thing, that is phase transition, not decomposition or degradation. So this is the single crystal. We have fortunately got two single crystal, one with PFD FA and one with PFD trimesic acid, that is TA. So this is the crystal structure. If you say in PFD FA, if you see that is 2 moles of drug, 2 molecules of, this is one drug molecule, this is one drug molecule, and basically fumaric acid forming a bridge between 2 drug molecules, and they are forming a new crystal, right? So this is the fumaric acid form. So if we do the XRD, now what we have done, because we need to prepare the formulation, so our aim is to prepare to bulk amount. Bulk means at least per batch, if we can produce 500 mg, 1 gram, then it will be useful for us. So what we have tried, that we have tried for the mechanochemical synthesis. So in mechanochemical synthesis, grinding time is very important, right? So what we have tried to say, after getting this, that is the simulated pattern. That means if you get the single crystal values, so single crystal, and from single crystal you will get one simulated XRD pattern, right? Simulated XRD pattern. So simulated XRD pattern, that is known as basically simulated XRD form, which we will get it from the single crystal data. So then what we have tried to do, that minimum time required to get the co-crystal by milling. So what we have tried by 1 minute, 5 minutes, 10 minutes, 15 minutes, 30 minutes. And if you see that within 1 minute, if we grind it, terphenadone and fumaric acid, they will form a co-crystal. And it's upon milling, it won't change its Bragg's diffraction peak. That means these peaks are retained. And this is quite similar as compared to the simulated pattern, so that we can say the crystal lattice of the single crystal and what we have prepared through mechanochemical synthesis, both are same. So now we have done some Mediated study, because we know that if any molecule participated in the hydrogen bonding, so either there will be a blue shift or red shift. So here, if you see this here, 1678, this wave number that has been changed to 16693. So that means there might be a red shifting. So there might be some interactions between these two, in this functional group, basically. So now we have done the property evaluation study. First we have done the equilibrium solubility study. Equilibrium solubility study we have studied in water, basically. So already, as I have stated, that pirfenidone solubility is high. It is approximately 20. Here exact value is 18.29 mg per ml. So we have studied 4 different co-crystals, basically 2 different co-crystals prepared by 2 methods. So we got 4 different co-crystals. So SC means by solution crystallization, that means evaporative crystallization. Lag means liquid assisted grinding, that is basically the mechanochemically synthesized. So for FA and TA, if we see that both from solution crystallization and liquid assisted grinding, we are getting almost the same solubility value. That means mostly 60% we got the reduced value. And in case of TA, it is very, very low. It is approximately 0.75 to 0.78. Now, we have studied the intrinsic dissolution rate study. Why it is important? Intrinsic dissolution rate study is nothing but the dissolution study from a single surface of entirely or exclusively the drug molecules. That means we would like to check the impact of new crystal on its drug dissolution property, because when we will prepare some sort of tablets, we will be putting some excipients. So that excipient might have some effect on dissolution, but we do not want to get this effect. We would like to get the exclusive effect of our change in crystal lattice, right? So that is basically, there is one ideal setup is available. So there you need to put some drugs or co-crystals, whatever it is. Then you will have to compress it. That means pellets you will have to form, and all the surface will be in a chamber. So only the exposed surface will be single, only one surface will be exposed. And from there we have calculated the cumulative amount of drug dissolved, right? And that has been plotted against time. So the linear portion, if we take any linear portion and if we take the slope of this thing, that slope is basically known as the intrinsic dissolution rate. That is mg per centimeter square per second or per time or whatever it is, per hour. So if intrinsic dissolution rate is reduced as compared to the drug, then we may assume that might be giving the sustained effect. So this is the ideal value and it is apparent solubility value. So now this is one theoretical calculation of ideal solubility. Ideal solubility will give you the information about the contribution of crystal lattice energy towards its solubility. That means ideal solubility is the solubility of a solute without having solvent, right? This is ideal solubility, all we have studied in physical pharmaceutics. That ideal solubility we can calculate from the basically DSC thermogram, right? So from DSC thermogram, there is one formula. So by using this formula, we can calculate the ideal solubility. It is in mole fraction unit. We can convert it into molality also if we have some solvent, right? Now, definitely we need to check the solubility, the effect of pH on solubility. Because our body is having a different gradient of pH from pH 1.2 to 4.5, 6.8, 7.4. So what we have studied at all the physiological pH, we have started the solubility study, and it was observed that there is no significant difference at different pH. That means we can ensure that whatever the co-crystal we got it, so that do not or doesn't possess the pH dependent solubility. So this is the kinetic solubility means what? That is kinetic solubility means at a different time interval, what is the solubility? So we have studied up to 24 hours because our aim is to get the sustained effect of the co-crystal. So therefore we have studied up to 24 hours and we could observe from this graph that it is almost similar, no significant difference was observed. So now any solubility study in case of co-crystal, if you would like to try to check that whether it is in solution it is stable or it is metastable or unstable. So only one thing we can do, that whether the residue is in which form. So from the solubility study we have collected the residue and we have gone for the XRD data. So upon seeing the XRD data, what we have observed that it is stable in all the pHs. That means co-crystal, that means in solution also co-crystal is the saturated species. And therefore, the residue is also a co-crystal. That means it is showing the similar Bragg's diffraction as of the co-crystal. So therefore, we can say its solution mediated phase transformation or solution stability is good. That is basically the solution mediated phase transformation study, which is most important for the formulation perspective. Right. So then we have studied the permeability based upon the Franz diffusion cell. We have studied the permeability also. So it is observed that in permeability also we got the that is this type of things we have got, that is a similar thing, right? So next it is our formulation work. That means how to prepare the formulation out of this thing. So for that we need to do the compressibility study, tabletability study and compactability study. So after doing all these things, we have ensured that we can prepare the tablet. And after that, we got the tablet and we have prepared doing all the excipient compatibility studies by powder XRD. And then this thing, DSC, we have confirmed that there is no co-crystal excipient incompatibility. So therefore, we have prepared the tablets. First, we have tried to prepare the 200 mg tablets by taking the equivalent amount of co-crystal, and then we have prepared the 600 mg tablets by incorporating the equivalent amount of the co-crystal. So these are the, all of you know, this is the predictive kinetic models. So we have done the dissolution study. So from there, from the Crossmeyer-Pepper's model, it was observed that in most cases, the dissolution mechanism is basically noise Whitney, that means Fick's law of diffusion. So that was actually our hypothesis, that if we can reduce the solubility, it will be converted into Fickian diffusion. So it is also obeys this model, because N value, if it is less than 0.4 or 0.45 in case of cylinder, all these things. So it is nearly about 0.4, 0.5 within this thing. So we can say it is following the Fickian diffusion. So what after that, what we do, we have studied the in vitro Raleigh study. Then we have studied, actually I'm skipping all these things, we have performed the toxicity study in animals. There was no significant, no abnormality was observed. After that, we have conducted the human biostudy, because as it is sustained release dosage form, so it will be very difficult to do this study in animals, because we need to convert into animal equivalent dose, and after that we need to put it in an intact form to the animal. That would be a very difficult task. So we prefer to do in a human study. So for that, we have studied the subchronic oral toxicity study. After that, we have studied the two study. Actually, one is single dose study, one is multiple dose study. That means whether our 600 mg once daily dose will be bioequivalent with the 200 mg thrice daily. So that was actually our purpose. So we have done this thing. So these are the pharmacokinetic. If we see this plasma concentration versus time profile. So this is basically single dose study. If you see, this is our reference formulation. We have taken the reference formulation, that is Perfinex from Cipla 200 mg, and this is our developed product, because if it is sustained, so its Tmax will be deferred, right? So it is from the graph, if you see, it is Tmax is deferred, although its elimination half-life is remains same. But after getting the fruitful results from the single phase study, we have conducted the multiple dose study. That means in a day, we have given 200 mg dose in morning, after lunch and evening. And same group, after washout period, we have given the 600 mg once daily. So if we see this line, it is giving the test formulation, that means 600 mg once daily. So it is giving this thing, and this thing we have given here, that means after 6 hours, we have given morning, after 6 hours, and again after 6 hours we have given. So it is showing this thing. So if we see all these pharmacokinetic parameters, so it was observed that only one parameter, it is basically, it is showing that entire portion, it is showing this thing, because this value is nearly same value, right? So once daily 600 mg pirfenidone fumaric acid co-crystal will be bioequivalent with the 200 mg immediate release dosage form thrice daily. So that was actually our aim. So we have calculated all the statistical calculation to prove which one is, whether it is bioequivalent or not. Definitely Cmax and AUC02T. AUC02T is bioequivalent because it is 0.8 to 1.25 values. And AUC0 to infinity also within this thing, but Cmax is not within this thing, although its log transform value is within the limit of the bioequivalence range. So over the summary, if we just summarize this thing, so we can summarize in that way that 600 mg pirfenidone fumaric acid co-crystal, once daily, it is bioequivalent to the 200 mg pirfenidone tablet thrice daily. That means in a day, we will take the 3 tablets And instead of that, if we take 600 mg once daily, so that will suffice the purpose. So this is the summary. So out of this, these are the some few references. So out of this work, we got two good publications. One is in crystal growth and design, another one is molecular pharmaceutics. So this is my acknowledgement, because this work is a part of basically PhD work of one of my PhD students, Nimmi Kumari. She has been awarded PhD, and now these are the students, Parag, Nupur, Srividya, Amit, they are working under me. This is my lab, right? So thank you. If you have any questions, I will be happy to answer those questions. Hello, can you hear me?
Yes, sir. Yes, sir. You are audible.
Please.
Yes, sir. You are audible, sir.
Yes.
So I would like to request the participants, if you have any questions, so please put your questions in the chat box, and also who are following us from the YouTube, you can put your question in the YouTube chat box as well. We'll take out the question from them and deliver it to the speaker. Sir,
If the drug is ionizable, how can you
if
differentiate drug
the
excipient?
drug is ionizable, how can you differentiate the drug excipient?
So I'm sorry that first I should say this thing, that which type of drug is suitable for co-crystallization, right? So here the best way to tailor or to increase the solubility is the salt formation, as all of us know all these things. But if the drug molecule do not possess any ionizable group, right, or they will not participate in the proton transfer process, then in that particular case, actually co-crystal will be the beautiful concept by means of which we can tailor the physicochemical property. So how can you differentiate drug excipient salt formation than the co-crystal? Actually, first of all, excipient we generally say here, that is actually we are using the second molecule that is termed as co-former, right? So if there is some proton, if you get, say, it is very simple thing. If you get the single crystal, is it co-crystal or is it salt? You will get it simple click, right? You will have to click it, then you will get the answer whether it is that drug molecule is participated in the proton transfer. If it is participated in the proton transfer, that is salt. If it is if it doesn't participate, and if it is formed through hydrogen bonding, that will be the co-crystal. So the best way to detect whether it is salt or co-crystal, it's means of by single crystal, right? That is a very simple thing. But in other cases, from the drug molecule, also from the chemistry point of view, if you analyze this thing, that there might be a chance of proton transfer, then also you can get this thing. And moreover, if you do the NMR study also, then also you can get the information that whether it is salt or whether it is co-former.
Sir, there's another question from Dr. Kiran Kumar. What is the effect of solvent in the formation of co-crystals?
What is effect of solvent in the formation of co-crystal? It's a huge effect, right? Just a minute. Stop sharing. Oh, have you stopped my sharing, right?
Yes, sir. Yes, sir.
Okay, thank you very much. Thank you. So, because if you see that simple, all of we basically perform the crystallization in our UG labs, right? So if you understand this thing, that effect of solvent on crystallization is same as of effect of solvent on co-crystallization. That is nothing. So if we change the solvent, there might be a different formation, stoichiometry will be different, or there might be a chance to not to form any crystal. So the solvent selection is very important. So depending upon the solvent, that means actually we generally prefer to choose a solvent in which both drug and co-former have a comparable solubility. Understand what I'm trying to say? That means the solubility of drug and co-former in a particular solvent, if it is comparable, that means it is somehow similar, then that particular solvent will be suitable for the co-crystallization basically. Sir, is it applicable to the herbal extract? It is very difficult to say because herbal extract, we don't know that what is their chemistry because it is not a single molecule. So if we do not have a single molecule, it is very difficult to say that whether it will participate in the. There are few reports where they are also trying to do with herbal extract, but it is not, the mechanism is not direct, it is established yet, right?
Sir, there is another question from our own faculty, Shormi Thapal.
Can you hear? Please.
Yes, sir.
Yeah.
So she's asking that how Raman spectroscopy may help to characterize the co-crystals.
Right. So Raman spectroscopy, actually, we know that if there is some non-covalent interactions of any functional group, so the Raman spectra will shift. Either it will be blue shift or red shift, right? So from there, actually, these FTIR, DSC, TGA, then Raman spectroscopy, all are supporting characterization technique of co-crystal. The best way to characterize is a single crystal and followed by powder excited. So now we need to prove it. Okay, say for example, we do not get the single crystal. Then it is very hard to prove that whether the new solid is co-crystal or some hydrates or some salt. What is this? It is very difficult because if you can get the different powder excited pattern, but that doesn't give you the information whether it is salt or co-crystal, which Souvik, I know him because Souvik asked, right? That you cannot predict this thing until unless if you do some NMR study, rigorous NMR study in solid state also. Because co-crystal, once if you put it in the solution, it may be dissociated. So then again you won't get anything. So in solid state you need to do the NMR study, all these things, right? So you don't call me Dr. Animes. It is better, Animes is better. Thank you.
Okay, sir. Thank you. If anybody has any more question, you can put the question in the chat box. Sir, I think there's no more questions. I would like to thank you from the Department of Pharmaceutical Technology for giving your precious time to us in this FDP of day 4. Sir, we have very small token of appreciation for giving your time, giving your knowledge to us. I would like to show that with your permission. We have a very small certificate for you to appreciate your time, to appreciate your knowledge that you have shared with us. And very soon after the puja, I'll be sending that to you in hard copy as well.
Thank you.
Thank you so very much, sir,
Thank you
for your
much.
kind participation.
No, thank you. Thank you. I'm leaving, okay? Hello, I'm leaving.
Okay.
Okay. Thank you very much. Thank you all.
Yes, sir. Yes, sir. Yes, sir. Yes, sir. So now we'll be moving forward to the next session, next part of our FDP. We already have our eminent eminent speaker with us. So I would like to hand over the session. So sorry for the technical problems. So we are already having our eminent speaker with us. I would like to hand over the session to one of our colleagues, Dr. Shonkita Das, the associate professor of Department of Pharmaceutical Technology. So, ma'am, kindly.
Hello, am I audible?
Yes, you are.
Good afternoon, everyone. Thank you, Shakur. Good afternoon, everyone. Today we have already reached at day 4, and it is the second session. Now, I, Dr. Sushmita Das, an associate professor of JIS University, would like to introduce our eminent speaker, Dr. Girishati Mohana. He has been working on molecular genetics or plant cellular and molecular biology for last 20 years. Presently, he is working as an assistant professor at R.V. Ramanujan Birla Institute of Technology and Sciences, Pilani, Hyderabad campus, India. After completion of his Bachelor of Science and Master of Science from University of Agricultural Science, he received his PhD from Ohio State University, US. He did his postdoctoral research at Ohio State University and Howard Hughes Medical Institute, Department of Biology, Molecular and Cellular Biochemistry, USA. Currently, his lab is working on epigenetics of megabase scale Rna gene regulation and atomic instability of rDNA and telomeres. He is also working on eliminate domain-specific functions of histone deacetylase in gene regulation and genomic instability, and to develop a novel non-transgenic epigenetic mutagenesis tool for plant improvement via HBAC modulation. This is a very small introduction of Dr. Girisha. Now I am handing over the session to Dr. Girisha. Sir, over to you.
Okay, thank you. Can you hear me? Can you guys hear me? Hello? Can anyone please respond? Am I audible? Yes? No? Hello? Hello?
Sir?
Hello?
Yes, you are audible.
Okay, all right. Is my screen visible?
Yes, yes.
Okay. Thank you. I don't know, is Amin here? Thank you, Amin, for the invitation, and thank you for the introduction. I know you guys, I don't know whoever is there, and online conversations are not necessarily great. They cannot be as effective as physical presence, but sometimes it helps to listen to speakers who are from different places. So I know whoever is here, who has attended the previous talk, you guys have already 1 hour, and your, what do you say, attention is kind of not so great because it's already towards the end of the day. So keeping that in mind, I would take this as more like a a slow paced talk. So pay attention to them, and just get few things out of this talk. So I will not bombard too many slides. First of all, my main focus is on plant biology, but I do have one objective on oral cancer because we work on ribosomal RNA genes as one of the areas. Of course, in the recent past, I am we'll be spending a lot of time in rice and tomato for some epigenetic tools-based improvement. So the title says engineered M1 Gs ribosomes as anti-cancer tools. Okay, so ribosomes have been used as anti-cancer tools, at least as a modality, not necessarily at the clinical level. But we are using this from a different perspective. But it's almost like a new strategy we are trying. Okay, so before that, I want to tell you a few things here. Let me just see if I can minimize this. Okay, great. This is, if you see in the textbook, people still use the word what is called as central dogma. I'm not sure if we should use the word dogma anymore. It is not dogma anymore. There are thousands of papers who have published that from DNA you get RNA and then protein, right? So there are several ways you can modulate gene expression, right? And there are several drug targets have been designed to either to target DNA, RNA, or protein, right? For example, if you take cancer, there are many cancer therapies, chemotherapies that target DNA replication, for example. Okay, so this slide is just to give you an idea of I'm sure most of you are probably aware. This, the basic tenets you can call our central theme. From DNA, you get transcription to RNA, and some of RNA becomes protein, and usually that is mRNA. Some RNA own, like tRNA, ribosomal RNA, and non-coding RNA, and they have their own functions, right? So now, whenever people want to knock down a gene or knock out a gene, they target the DNA or RNA or the protein. Okay, so what I'm going to talk today is about RNA cleaving tool. Okay, so in a way, it's a down-regulating genes or knockdown strategy, you can say, or it's also called as a reverse genetic tool. Okay, oops, it's not moving. Huh? What's happening? Okay, so if you talk about the global cancer statistics, abnormal cells proliferate uncontrollably and invade adjacent regions of the body, means that is metastasis. By now, everyone knows of the cancer. And I believe as much as we are doing research and so much we are stressing ourselves in doing research and getting cancer, perhaps, on one hand, we are stressing ourselves to say that we study cancer, but on the other hand, those stress itself can have an impact. Okay, so one has to really understand our own biology. That's also very important, because in the name of science, in the name of whatever the career success, if you put a lot of stress on your body, it cannot take, although you are studying cancer, right? So that's something kind of, caught my attention when I saw people, be it in US or in India, they do research and they do publish things and they say, well, your own research says stress affects or this affects, but why are you not taking care of yourself? I mean, is our research and teachings for someone on the aliens, or is it for human beings, right? So that is something you all have to pay attention in. At least I have benefited from my own understanding of biology. Let me make that statement at the beginning. So, there are several cases, several deaths. It's not surprising. And in our lab, it's, as I said, it's one of the objectives. Of course, I do in collaboration with my colleagues who are cancer biologists. I myself not necessarily a cancer biologist. As I said, I'm a plant biologist, with major focus on epigenetics and chromatin biology and molecular breeding, whatever. But when you study something as a model system, that's how you get trained in US. You can work on any other system if one of your objectives overlap. Okay, and so there is. You have to just give me 1 second. Hello? Yeah? Yeah.
Sir, I forwarded, sir.
Okay, thank you.
Okay, sir.
Oh, I'm sorry, I have to take that call. Important. I know it was only 1 second saying yes or no. Something has to be sent today. I apologize for that. Where is that? Okay. So this head and neck squamous carcinoma versus HNCC is called, world's 6th most prevalent cancer. And whatever the numbers I have given, 0.3 million new cases or 0.2 million deaths. But I think it's one of the what you say, India accounts for the highest number of oral cancer cases because you can see there is a lot of tobacco chewing in India, right? Unlike other countries. Of course, given our population, probably we rank top among 10, 5 in many diseases because, perhaps, we have been now becoming capitals of many diseases, it looks like, all right? So then what is this connection of ribosome biogenesis to cancer? So you all have studied in biology, ribosomes are organelles, both in prokaryotes and eukaryotes that synthesize proteins, correct? So they are, although ribosomes synthesize proteins, if you have paid careful attention, you will realize that it is the ribosomal RNA that acts as the catalytic components of ribosomes. Means joining amino acids, forming the peptide bond is done by RNA, not proteins. Proteins are needed, and that's why ribosomes can also be called as ribozymes, and that's a different. I'm not talking about that ribozymes today, but conceptually ribosomes are also ribozymes. I'm just showing you a quick animation. These ribosomal RNA genes are present in hundreds of repeats, one next to each other, both in several eukaryotes, and they have different structural ribosomal RNA. Now you might have read about these things: 18S, 5.S, 28S. So RNA polymerase 1 transcribes them, and it first generates the whole transcript. It's called 45S pre-rRNA. The 45 is based on the sedimentation value, right? If it is smaller than, some organisms have 35S, in some cases it is 47S, that could vary, but by and large, this is what happens. RNA polymerase 1 transcribes, and then, unlike mRNA splicing, here this RNA undergoes processing. Means you have to cut things what you don't want, just like how you cut the vegetables, right? What you don't want, you cut them off and use whatever you want. This is pretty much what happens. So you get 18S, 5.S, 28S in humans, and there is another ribosomal RNA which is 5S that is transcribed by RNA polymerase 3, means by a different polymerase, and they all come together, make large subunit and small subunit, right? And then protein synthesis takes place with the help of ribosomes. And these ribosomes have several protein subunits, somewhere around 80 to 100. Okay. This is and these ribosomal RNA genes are clustered in 5 different chromosomes. Means, as I said, about 300 copies of these genes, and they all occur as clusters, means repeats one next to each other, and they are located on 5 different chromosomes of humans. That is on chromosome number 13, 14, 21, 22, as I listed in this slide. Okay. It has been observed that 45S rRNA genes are dysregulated in human cancers. Means they're actually upregulated in several human cancers. If you see those findings, that itself is not very surprising, because you know that cancer means uncontrolled cell divisions. You need more proteins. More proteins means you need more ribosomal machinery. Therefore, it makes sense why these genes are upregulated in several human cancers. All right, colorectal cancer. I'm just giving you some example. Multiple cancer types. Oral cancer was studied very recently, and ours is one of the study which will be published very soon in Cell Lines. Okay. Then whenever someone talks about the gene regulation, upregulation or a downregulation, they ask one question: is it controlled by, or is it contributed by promoter DNA methylation status changes? So the studies, what they have reported, in some cases it is linked to hypomethylation of DNA promoters. In some cases it is not linked. Okay, so means there are multiple ways these genes can be upregulated, is what we can figure it out from. So as I told you, why they are upregulated, because to synthesize effectively enough cellular components which are made up, many of them are made up of proteins. All right, and all RNAs are essential component of ribosomes. Okay, that, as I told you, it makes sense why you need a lot of them. Okay, I hope so far I am clear in what I have said. Okay, and then comes some cancer biologists ask this question: hey, look, in cancers ribosomal RNA is upregulated. Can we target this transcription somehow and bring down the ribosomal RNA levels, and can we see if that helps to reduce the cancer or treat cancer? The answer turned out to be yes. Okay, and they have come up with chemical inhibitors which inhibits the RNA polymerase 1. Okay, there are 3 examples I'm putting here. One is, and I'm also mentioned the authors which have made this chemical or tested this chemical, designed this chemical. It's called CX5461. Means they come up with some names. They don't exactly tell the chemistry unless they get the patent or whatever it is. But the bottom line is, when you see here, this basically, see, try to simplify this talk. Okay, don't complicate. Try to understand all the components. Basically, you are blocking the RNA polymerase 1 transcription. Means what? Ribosomal RNA is reduced. So why I'm showing 3 different examples is they have 3 different compounds. They're targeting different components, right? The drugs can be targeting different components to achieve the same effect. So same thing here. Okay. So then people also have tried other models like you can, it is, if you guys are into molecular biology, that small RNA technology also has been used, which can be used to downregulate the genes, any genes for that matter. siRNAs are commonly used, which induces double-stranded RNA-based gene silencing. It's also called RNA silencing. Okay, it's also called RNA interference. But in this context, we are exploring an engineered bacterial ribosomes called M1 Gs as a novel anti-cancer modality. And this we are trying in collaboration with Professor Venkat Gopalan lab at the Ohio State University. I worked in this lab for about 15 months, and he was also my PhD thesis committee member at Ohio State University. So then comes, what is this M1 Gs ribosome? Okay, the concept is fairly simple. So try to just get what exactly it does. Really simple to understand. Other complexities for the time being you ignore. Okay. This is called ribonuclease P. Ribonuclease P is a ribozyme, means RNA acting as enzyme, correct? So it's there both in prokaryotes and eukaryotes. What is its function, or who discovered it? And this was discovered by Sidney Altman, wherein the first time they showed that RNA can also act as enzyme, and which are called ribosomes. That's why it was a major discovery in biology. He received Nobel Prize in chemistry for this. So what is this enzyme? This is a ribonucleoprotein complex. In E. coli, it has M1 RNA. It's a catalytic RNA, and it has one protein subunit. But which among these two will do the catalysis part? It is the RNA. That's why it's called ribosome. Right? Now this is the structure of M1 RNA. Okay, so this is how it looks with the folding. So you don't have to remember it necessarily, but this is how it looks. So now what does this enzyme do? What is the function of this ribosome? Well, it functions in maturation of the 5' end of the precursor tRNA. So whenever you ask this question, why are RNAs, like tRNA is made, why don't you make tRNA exactly you want? But that's not how the organisms are making. Perhaps they may have a reason. They make it something extra, right? It's very similar to, you go to a shop, you need a 1 meter wire. So do you get 1 meter wire? No, usually get something extra. And afterwards, if you want, you can cut with scissors. I think biology is also means the cellular biology is also following similar principles. Have something extra, let's later on we'll trim it, So something like that, you can see this is a tRNA. And where I put the arrow mark, I'm hoping you can see my pointer. All right. This part, what I'm pointing with laser point, this is extra. That needs to be removed before this tRNA can become mature tRNA or functional tRNA. And someone has to cleave this off. Who will cleave? RNase P. RNase P is a ribozyme, means RNA acting as an enzyme. It cleaves here. So that is what it does. You can see this is a pre-tRNA. And I can see I have animated the RNase P acts here and cuts it. See, it's like it cuts, right? I show it that animation. That's what its function is. All right. So this is mature tRNA. However, Sidney Altman group earlier, a couple of decades ago, thought, can we convert this into a tool of gene targeting strategy? Okay. So for that, they found that it recognizes the structure, not the sequence. Means anything that looks like the structure, RNase can recognize. So that was an important find. Okay. It's like, kids, whenever somebody is wearing something, and if they think it's parents, anybody who's wearing that color dress on that day, they think that it is their parent when they're babies, right? They just go by some color or maybe some shape or something like that. So here, its recognition is based on the structure they found, not based on the RNA sequence. So based on that, they found this tool called custom designed external guides. What does it mean? Well, you assume that you want to target an RNA encoded by some gene, correct? So you take that sequence and make it which is complementary to this sequence, right? So your red part is your target RNA, then its base pairs with this. See, when the target RNA base pairs with what is called as this external guide sequence, this blue and green part, can you guys now see it looks like tRNA? Yeah, actually it is not tRNA. You have providing this to the cell. This you are expressing, correct? But it hybridizes to your target RNA. How? Based on base pairing. Now, because of this pairing of your target RNA and the so-called external guide sequence, this looks like tRNA, and the RNase P that is there in the cell which cleaves here. What happens to the target RNA? Once you cleave at the 5' end, the RNA becomes very unstable. It gets chewed up easily, right? The function is achieved. Means you have designed an RNA which is called external guide sequence based on the target gene sequence, and it binds, and RNase P it believes that, oh, here is a tRNA, I'm going to cut it. So the target mRNA. So based on this, you can target whatever the mRNA you want, ribosomal, any RNA. You can target it using this strategy. Okay. This is first strategy. The another strategy is, I will tell you, but this strategy is called external guide sequence. Okay. There have been several studies published in really good papers like PNS, nucleic acid research. There are many. I am just quoting few which have used this to target the RNA, and one of them was to some oncogenic products, BCR ABL. There they found that somehow the mRNA is getting joined, so they used this to cut it like a scissors, right? So I am hoping I'm audible and at least some of you are following. You know, I hope more, but you know, at least a couple of them, even if you are following it, at least the purpose will be solved today. Now let's come to what we're talking about: engineered M1 RNA, right? M1 GS as a tool to cleave specific RNA. Here the strategy is little different. They're not using external guide sequence. They used this bacterial M1 RNA. I told you this is the ribosome of E. coli, correct? Now, and the green part is the sequence you want to target. So whatever the sequence you want to target, you choose that region. There are some features you have to decide, and you fuse that sequence to your M1 RNA, correct? Means your M1 RNA has guide sequence. What is this guide sequence? Basically, this is complementary to whatever the target RNA you want to target. That's why guide sequence is not fixed. M1 RNA is fixed, and the end part ACCA is fixed, but what changes is guide sequence. It changes based on what? Based on whatever the RNA you want to target. So when once this guide sequence matches with this is good enough for mRNA to recognize. They found, oh, you know, you don't need the whole tRNA structure, just it's called a stem structure, right? Stem structure is good enough for this ribosome to detect and cleave it. So here I've shown you as a scissor symbol, it cleaves here. RNA is degraded. So when someone designed this, they also designed a mutant M1 RNA. Okay, means it is called inactive mRNA. So why do you need that? You need this as a control, right? How do you tell that once you express the mRNA in a cell, this effect is only due to this? It could be due to something else. So that's why, to prove that it is actually whatever effects you are seeing is due to this RNA, you have to always use some control which is inactive, correct? So there is inactive M1 RNA available. We have both of these constructs, which has been given by Professor Gopalan lab. Right now we are using it. Okay. So again, summarizing this, what is this engineered M1 ribosome? It is a ribosome to which you join the guide sequence. That's all. And that guide sequence is complemented to any target mRNA. And once they bind, cleavage happens. All right. Of course, if you use the inactive M1 RNA as a control, then cleavage does not happen. So binding takes place, but no cleavage, because this catalytic activity has been removed from this mutant, and therefore it is used as a negative control, right? So why use M1GS when RNAi can do the job? Means there is Rnai strategy also. So we found there are some advantages to this tool. This tool is highly specific as per the literature. When you say the RNA mechanism, there are so many components involved, several genes involved. Can you overexpress all of them? It's not easy. Whereas in M1GS, you have only one construct to be overexpressed, right? So remember, whenever you want to modulate gene expressions, one gene is easy compared to several genes, right? So that's how simple it is. And also they found that when you express these M1GS constructs in human cells, up to 40 days, they would not lead to any cytotoxicity, right? This is an important point. I'm sure many of in pharmacy, you must be very familiar with this, that any drug which leads to cytotoxicity is not good, particularly if it is for the humans, right? So it should be tolerable, right? And M1GS ribosome is an exogenous system and does not depend on the endogenous system for its function, okay? And nor does it affect or compete with other endogenous systems like mRNA. So this is external. Exogenous means externally you are putting it one, and it does not depend on any endogenous components. As I said, if depends on endogenous components, then they also have to be more to function. And if there is a deficiency of any one of the internal components, then your system will not work if it depends on it, right? For example, if you are going and giving a lecture in a classroom, you don't depend on anybody, you can do effectively. But let's say you are depending on some 10 people to help you out, and if one of them is not there, you can't deliver whatever you want. It's as simple as that, if I want to give a simple analogy, right? And of course, M1 requires 12 hours to result in a significant prediction, and whereas RNA resides like 24 hours. Probably sometimes this time difference may not be a big deal, okay? But there are few advantages. The bottom line is we thought that this M1 strategy can offer some advantages over the other systems. Coming back to the question, M1GS strategies to downregulate RNA in cancer. Remember what did I said in the beginning? In several cancers, ribosomal RNA are more expressed, means there is abundance of ribosomal RNA. Someone asked a question, some scientists, cancer biologists said, hey, can you bring down the ribosomal RNA and control the cancer? They found inhibitors worked well. Now, along those lines, but we are trying a new strategy to bring down the ribosomal RNA, right? At least as a proof of concept. So for which we are trying to use M1 in ribozymes, right? Design a guide sequence specific to ribosomal RNA and cleave it. Basically, we are using it as a scissors, right? Using it as a scissors to go and cut the ribosomal. Very as simple as that. But of course, we have to prove that it happens, and it will bring down the, it will have the anti-cancer effects. So what we do is, earlier I mentioned in my introductory slide, rDNA transcription means ribosomal RNA genes are transcribed by RNA polymerase 1 in eukaryotes. Okay, eukaryotes have multiple RNA polymerases. Prokaryotes also only one. So in eukaryotes, RNA polymerase 1 does this job, all right? And we want to express M1GS under using, so that anytime you want to express a gene, you have to use a promoter. So which promoter you would use? So we propose to use the promoter of RNA polymerase 1. Why? Because this promoter is highly active in cancers. That's why you see a lot of ribosomal RNA getting expressed. So use ribosomal RNA promoter, you want M1GS ribosome. So you express more of this ribozyme, right? Which has the guide sequence to target ribosomal RNA. It's more like a scissors. Imagine that you are preparing a scissor which is very active to cut, basic. So diagrammatically showing, you can say this is RNA pol1 promoter. This is the customized M1GS to target ribosomal RNA. Because of this promoter, you will be making more of these scissors. That's all. Okay. Therefore, we expect levels of M1GS and rRNA to be much higher in cancer cells compared to normal cells. So what is the advantage? What is the expectation whenever you design any drugs, like for example, chemotherapy for cancers? So does it not have side effects? There's hardly any drug that does not have side effects, right? You guys probably know better than many others. So the idea is to minimize the side effects, because this M1GS, if it starts cleaving the ribosomal RNA in the normal cells or non-cancer cells, then you are killing them. So here we are hypothesizing in cancer cells, ribosomal RNA is already expressed more compared to normal cells. So point number one. Point number two, your expression of M1 just will be high in cancers because you are using its promoter compared to the normal cells. Means the enzyme is more, substrate is more in the cancer cells compared to normal cells. Therefore, cleavage will happen more in the cancer cells compared to normal cells is our idea. Okay, so we have to test this idea. So conceptually, that's why I said we expect more cutting in the cancer cells compared to normal. So this has been diagrammatically shown here. Let's say what happens to the expression of ribosomal RNA. It has been already known, right? I mentioned it is a lot of ribosomal RNA is made in cancer cells compared to normal cells. These threads are like ribosomal RNA. And you express this M1 GS using the promoter. So again, this is a ribozyme, right, with the guide sequence. As you can see, there is a enzyme is also, ribosome is also expressed high. We expect it to be high in cancer cells compared to the normal cells. And therefore, the cleavage, what we are saying, cleavage also should be more in cancers compared to. As a consequence of all these things, frequency of rRNA mediated downregulate, this rRNA downregulation mediated cell death means you don't want normal cells to die in big number compared to cancer cells. So we believe, or we hypothesize, probably this could happen in our strategy, right? Ideal. So as I said, we need to test that. So now, of course, we can't just go and apply this tool into the oral cancers or whatever it's called, head and neck cancers. We first need to test whether ribosomal RNA genes are upregulated in oral cancers. Although it has been shown for several cancers, these genes are upregulated, a lot of ribosomal RNA, but we can't take it for granted, right? So first we took some multiple cell lines, cancer cell lines, oral head and neck cancer cell lines. These are the numbers given. There are numbers SSC25 taken from tongue, Cal27 taken from tongue, and FADU taken from pharynx. And we have some non-cancer cell found at 1A, which is isolated from esophagus. We used 3 biological replicates and technical replicates within as we had to do. This one was done within collaboration with my friends here, Dr. Piyush Kandelia and Vivek Sharma. And Dr. Piyush has provided the cell lines for us to study these things. So what was the idea? You grow these cell lines and ask one question. What is that? Are ribosomal RNA expressed highly in these cancers cell lines like any other cancers? To our surprise, we found that these genes are downregulated, which was a surprising result for us. With it, we first, when we did one experiment, we thought, oh, is it something happened? Let's repeat it. Yeah, our students repeated it, and we found the answer is true. These genes are downregulated. So we studied multiple ribosomal RNA. Like one is what is called as pre-45 sRNA, means whatever gets transcribed, first before processing takes place. Then 5' sRNA levels we measured, 28 sRNA levels measured, 18S we measured. All these are transcribed by RNA polymerase 1. 5S ribosomal RNA also we measured, which is transcribed by RNA polymerase 3. So what is the result? In the cancer cell lines we studied, ribosomal RNA is not upregulated, but it is downregulated. Then we thought, wow, because sometimes when you study cell lines, is it really true or is it making sense? Because how would you explain? Cancers are cancers. There is a cell division. You need more protein. Then now you are reducing the ribosomal RNA. How can you grow, right? So we can't explain this. But around this, we were about to get these results. Of course, this is just we used for the quantitative PCR or RT-PCR, and for that we use some recent estimate methods. That's not very important. What are the statistical methods? It was a pretty straightforward statistical analysis. We can clearly see using the bar graphs that they are downregulated highly significantly. But around the same time, a Chinese study are also published, and that was in a oral cancer tissues. They said RNA genes are indeed downregulated. Then we said, oh, whatever we found is probably true. Probably is what is happening in the oral cancers. still it does not, they also have no explanation. How would you make sense out of this when these genes are needed more and it is not there, right? It is like, you are going to, you are arranging a function for, arranging a lunch for 1,000 people, and the ration you are not ordering much. Okay. So how do you make food for so many people, right? Of course, there could be other mechanisms how it is managed, but this is just the beginning of the story, right? Because remember, they have published only in 2019, couple of years ago, and now recently our finding, we are now about to publish our results, because we were carrying out whether DNA hypermethylation is happening or not. Okay. So all the details. The bottom line is ribosomal RNA genes are downregulated. Therefore, we cannot apply our M1 Gs strategy in these oral cancer cell lines. Correct. Of course, we will publish the data. Then we'll go back to some other cell lines, like where like a breast cancer cell line, prostate cancer cell lines, which are available in India also. Because we have to prove that, we have to show our tool as a proof of concept, right? We thought oral cancer is one of the leading problematic cancers in India. That's why people would like to fund to work on this cancer. Though we thought we will do that in oral cancer itself, but that there's a no scope for reducing the ribosomal RNA, because ribosomal RNAs are already reduced. But we will not stop here. So we'll probably pick the couple of other cancer cell lines, for which it has already been shown earlier that there are ribosomal RNA overexpression. There we can try to cleave them, right, using this ribozyme tool. So that is where we are heading. All right. Oops, what happened? All right. Looks like it got stuck. Okay. So I think, I hope you guys are, conclusions is that ribosomal RNA genes are downregulated in head and neck cancer cell lines and tissues, and its link to DNA methylation stages needs to be carried out. In our case, we recently completed, we haven't looked at the data yet, so we just got the data. So interestingly, it remains to be investigated how in these cancers, high demand for protein synthesis is met given the downregulation of RNAs. As I told you, how will you supply food to thousands of people when your ration is so less? Something like that you have to explain. Of course, as I said, just downregulation of RNA does not mean that downregulation of protein synthesis. So people have to really figure out what is going on here. At least two studies are telling that. One is ours and the other one is another Chinese study, and they have studied the actual tissues. Ours is at least a cell line model. So theirs is an in vivo model, right? So, and targeting RNA pol-1 transcription or 45S RNA may not be ideal therapeutic strategy to control head and neck cancers. That's why we said we can't target RNA pol-1, or anyone else can't think of targeting this as a anti-cancer therapeutic strategy. Okay. Oops, this is not moving. I don't know what's happening. Sorry guys, it is slow. Hello, come on. Oh my God, what's happening? I'm not able to. Let me just do something. Okay. M1 strategy can be explored as a novel anti-cancer modality in cancers. As I told, we will try other cancers where RNA genes are upregulated. But we are still interested in this ribosome strategy, but oral cancer is not the best thing to use, right? So that's all our kind of strategy and goal, and hopefully I'm able to make sense out of this, and hopefully you have got some of it at least. So as always, acknowledgements. Okay. So current funding, I'm luckily I have been able to receive funds from multiple funding agencies for other projects. For this one, I received mainly funding from our institutes. There is a Center for Human Disease Research Initiative, and I'm one of the investigators because of this work. And my student, PhD student Neha, received ICMR SRF to carry out this project. And of course, as Ramanujan Fellowship, also one of the objectives is to work on RNA gene regulation in cancers. And Neha also received some kind of prize, in the national conference competition on this work. Okay. And I'm trying to show, I don't know, it's kind of every now and then it's blocking. I don't know. All right. So in the acknowledgements, of course, the few people I want to acknowledge, so of course, our institute, and this is Professor Venkat Gopalan. He's a very good scientist, really. And he worked with Sidney Altman. Earlier I showed you he was the Nobel laureate. So this guy worked with him, and I worked with this guy. Really, it's nice to work with him. Learned a lot from him. A very disciplined scientist, very good person. So he has been helping me to carry out this project. Means he has given all the constructs, ribozymes we need, and we have developed those. We have kept those tools ready to go. And of course, my Department of Biological Sciences, and my colleagues here, Dr. Piyush, Dr. Vivek, and Dr. Swati Biswas. Actually, S is missing here, I'm sorry. And this project is mainly driven by students, and one is not here and is working as CSR JRF fellow in another institute, Naveen. And Neha, as I told you, she is a 5th year PhD student, and this all work is now done by these two guys mainly. And right now, Neha is continuing. And thank you for your attention. And I will try to answer questions if there are any. Thank you. Hello.
Hello, sir.
Thank
you, sir, for your valuable speech. If participant, is there any questions from your side? You can put your question in the chat box.
Okay, sir. I have two questions based on your slides and the presentations. Undoubtedly, it was really good. Actually, these are the two challenges even I was noticing during my PhD work, and it was really very.
Hello. I think we lost you.
Hello, can you hear me?
Yeah, now
Okay.
I can. Yeah, now you are back.
So, what I was mentioning that this topic, what you said about the gene targeting, right? The particular gene target, and you are cutting it from that your specified area of the sequence. So, the Cas9, everyone is trying to target the target sequence, but the major thing which we are having still now the limitation, that is the specificity of that particular sequence. Now this M1 GS gene targeting tool which you described today, will it be applicable also for the Cas9 machinery? Can we apply this concept there as well?
Yeah, see, CRISPR-Cas9 is also a gene targeting tool, right? That's also gene targeting tool. It can be used to cleave RNA also. There are and it can be also used to cut the DNA, wherein some mutations can happen, and that's how genome editing is done, right? So it can be basically, it is a RNA cleaving tool. You can try to target to any RNA you want, and so far specificity seems to be good here. Okay, but you have to understand one thing: the specificity, the guide sequence length. I said, right, because you have to tell M1 RNA to where to target, right? That is where you give the guide sequence. It could be 9 base pair, 10, 11, 12, 13, 14. As the length of the guide sequence increases, obviously specificity increases, right? Because 10 nucleotides you can randomly find anywhere in the genome compared to 50.
Yes, yes, yes.
But the caveat with you have to play with that condition, because as the specificity increases, it's good for you. But as the length increases, the M1 rhizome's cutting efficiency is also a little low, because it has to leave that and move on to next one. Right?
Yeah,
Because
it is leaving a tail behind. Yes, yes.
so now let's say you have guide sequence, it binds, correct? It cuts, because the binding is so strong, it will not leave quickly. Like that's why this M1, the scissors has to go and cut somewhere else, right?
Yes,
But
yes.
it is holding on to substrate a little longer. So it is a trade-off between your efficiency and the specificity. That's why you may want to try, like maybe try 10 nucleotide length and 13 nucleotide length and 15. And your off-target effect is really less, then you can settle with that guide sequence. There is no specific guide sequence mentioned in the literature saying that this is optimal, because it depends on your sequence also.
Yes, yes.
But specificity can be achieved. CRISPR-Cas9 also they're trying to improve that.
Yeah, in the CRISPR-Cas9 also the major limitation was in the space, because you are targeting from a gene, but it is also cutting the untargeted sequence.
They're also trying to improve the specificity, but it's still a problem, it seems.
Okay, so you think that is mainly due to the nucleotide base pair optimization? That will be one of the major part of the address.
Here, with respect to M1GS, at least,
with this your tool M1GS, you are saying it is more the specificity of the bind.
Specificity is more. Specificity has been shown to be high. One of the features, and as the literature says, one of the studies. And as I said, you can increase the specificity by increasing the length of the guide sequence.
Okay, and one more thing is you are talking about the ribosomal RNA, but now there is a very new the conception has tried with this similar kind of to shoot out the problem with the siRNAs. But I think in your presentation you more stressed on the ribosomal RNA specificity, but I think now the siRNAs are having really a very major role in this sequencing specificity to target, because they are very specifically binding and you can get the translated product. As you said, I like that question what you said, that there was a challenge that even if you downregulate the gene, that doesn't mean the protein is downregulated. There can be the protein maybe the same level. So but if you are targeting the siRNAs, I think this point can be well addressed.
No, but see, the idea is here, siRNA of which you will target, right? Because siRNA involved in some cell survival is altogether a different strategy, correct? So in this strategy, the ribosomal RNA themselves are targeted, correct? So the idea was that ribosomal RNAs seem to be abundantly expressed in cancers, correct? Of course, small RNA have been used to downregulate ribosomal RNA also. One of the tools I mentioned in my talk, but here the idea is to downregulate the RNA as a strategy. As I said, the different genes can be targeted to control cancers. Like people have done that. So here our focus is on to bring down the ribosomal RNA levels. There are multiple strategies people have tried, like a chemical inhibitors to inhibit the transcription. Small RNA have been tried. We proposed ribosome can has much better advantage compared to these, because siRNA you have to supply them. Here you can supply the DNA and DNA can make RNAs override. Actually, I could not go into those details. So there are so-called perceived benefits. We have to demonstrate that in our system. We are not saying, but why perceived benefits to downregulate the ribosomal RNA. But you have to see here, our idea was to downregulate the ribosomal RNA. That is our overall theme of the project.
Okay, thank you so much.
Participants, and thank you all. Now, on behalf of Department of Pharmaceutical Technology, JIS University, I want to display a certificate as a token of appreciation. Sir, can I share my screen with your permission?
Yeah, sure. I think I should stop sharing.
Sir, please accept it.
Yeah, thank you.
Thank you very much for your nice and valuable speech, sir.
Mhm.
Thank you, sir.
Thank you.
Happy Puja, sir.
Thank you. Thank you. So then I will leave the meeting. Okay.
Okay, sir.
Okay. Thank you.
Participants, we have already shared our feedback link. Please fill up the form and give your feedback. And from tomorrow is the last day of FDP. From 3 p.m. The session will be started. Please join on time. Thank you all. Thank you.